All right, so yeah, next up we have Dr. Shing Ho, professor in the Department of Biochemistry and Biophysics at Colorado State University in Fort Collins. Dr. Ho received his bachelor's from Franklin and Marshall in 1979 and his PhD at Northwestern University in 1984. He was American Cancer Society postdoctoral fellow at MIT from 1984 to 1987. And at that point, he started his faculty career, in fact, here at OSU in 1987, working his way to full professor and department chair. When in 2007, he joined the Colorado State Department of Biochemistry and Biophysics as chair there. Over his career, Dr. Ho has authored more than 90 peer-reviewed publications with over 10,000 collective citations. He's received countless awards for his exemplary research, including the NSF Director's Award for Superior Accomplishment and for his outstanding teaching accomplishments. Dr. Ho is famous for many things, including having solved, I think, the first structure of the DNA holiday junction, and also for discovering halogen bonds in biological systems. I would also argue that he was of great importance here at OSU because he was the person who sprinkled the first seeds of genetic code expansion in our department here. And that happened when, in 2007, when Shing was department chair, he was becoming increasingly interested in incorporating halogen bonds into proteins as a means to increase their stability and create new functions. So at that time, he got in contact with Ryan Mell, who was very early in his career, also at Franklin and Marshall at the time. And he asked Ryan if he could encode halogenated phenylalanine residues into lysozyme to effectively engineer the first protein with stabilizing halogen bonds. It just turns out that as Shing's last rotation student here at OSU, it was my rotation project to purify and crystallize those proteins that Ryan had made for Shing. And so we're here 17 years later with Shing having built a whole research program on the concept of halogen bonds in protein, and Ryan Mell now is GC for All director here. So it really is a small world in many ways. Anyways, Shing, it's a real honor to have you here, and I very much look forward to your talk. Take it away.
Great. Well, thank you for that introduction. Let me start by sharing my slides, and want to congratulate Chris on a very nice talk on their systems, on the various readers of trimethyl lysine systems. Anyway, so again, thank you for the invitation to give a talk. I'm going to talk today about something that we've been doing with genetic expansion. That is, we're trying to create actually new catalytic functions, or new catalytic centers in enzymes. And we're going to use something called halogen bonding to be able to do that. So my talk today, we're going to start off with a very quick really review and definition of what a halogen bond is. I'm sure not all of you have ever heard of a halogen bond. Then we're going to go to perhaps to connect back with Rick and Ryan at OSU, what we had done with trying to stabilize T4 lysozyme using protein engineering using genetic code expansion. But then I'll spend most of my time talking about this new project that we started about 2 years ago, in which we try to incorporate halogenated amino acids into developing and creating new enzymes that uses chemistry that currently doesn't exist in nature. Let me start with describing what a halogen bond is. In 2004, we wrote a paper that actually started to recognize that something called halogen bonds are important in biological molecules. And they're important both in terms of the binding of halogenated hormones such as tyrosine or thyroxine in your bodies. That is, if you look at thyroxine, thyroxine is an iodinated hormone, and the recognition of the iodination site or state of the hormone is dependent on the short interaction between, for example, an iodine and an oxygen within the protein itself. The other place we found that halogen bonds were important is also in other types of ligands, particularly inhibitors. In this particular case, this inhibitor against a protein kinase is dependent, again, on short interactions, in this case, between a bromine and oxygens of the protein backbone. And in fact, halogen bonding has become now recognized as one of the key elements in protein and in drug design against clinically important protein targets. So one question is, well, why is there halogen bonding? What are these things? If we think about general chemistry of halogens, halogens in the periodic table are considered to be well, highly electronegative, and therefore they harbor a negative charge. And the question is, why would these atoms that are effectively negatively charged interact with things that are electron rich, like oxygens? And the effect really can be described very simply by a simplistic electrostatic model called the Sigma Ho model. And in this model, what we see is that if we would take a halogen, such as this one, and covalently link it to a methyl group or the carbon of the methyl group, we would take the electrons in the valence states, pair them up, and what we end up doing is making a sigma molecular orbital or bonding orbital. In doing so, the electron that would be normally distributed across the pz orbitals of the halogen becomes subsumed into the sigma molecular orbital and effectively depopulating the pz orbital that's opposite of that sigma orbital. As a result, the effective radius or size of the halogen in the direction of the pz lobe becomes slightly smaller. And in addition, because of the lack of electron density there, would expose the positive charge of the nucleus so that we end up with a slight partial positive charge that we call the sigma hole, which is a positive crown. And it's this positive crown that would interact electrostatically, let's say with the keto oxygen of a protein backbone. If we look at this sigma hole, what the sigma hole model predicts is that iodines would have a greater halogen bonding potential than bromines, and that greater than chlorines, because the polarizability of the electrons of the halogens are going to basically scale proportionally to the size. That is, the larger the halogen is, the more polarizable it would be, and therefore the larger the sigma hole would be, and the stronger that halogen bonding potential would become. So one of the things that, as Rick said, one of the things that we wanted to do was to see if we can incorporate halogenated amino acids into proteins in order to affect their stability. The first system that we tried to do this with is with T4 lysozyme. And after many years, perhaps 2 decades of work, we were actually successful in doing this. We incorporated a meta-ionated tyrosine into this tight loop of T4 lysozyme. What we found is that the smaller of the halogens, which is chlorine, will actually be able to fit into this tight loop to form an interaction that geometrically is consistent with the halogen bond. And in doing so, ended up stabilizing the enzyme by about 1 degree Celsius, which is equivalent to about 3 kcals per mole of additional stability. Perhaps more interestingly, what that does is it takes this chlorinated enzyme and it actually ends up being about 15% more active at elevated temperatures than the wild type because of the additional thermal stability of the system. So after 2 decades, Rick, I can tell you that the experiment finally worked. Well, about 2 years ago, I was thinking about how to really renew my NSF grant, and over in night where I couldn't fall asleep, I thought of kind of an audacious experiment, and I proposed this to my a new graduate student, again, who was rotating in my lab. And I proposed this experiment where I said, I think we can take halogens, which are on the right-hand side of the periodic table, and replace the functionality of, let's say, a magnesium, which is on the opposite side of the periodic table. And basically the idea is that we can take the properties of the halogen bond, potentially the electrostatic components of the halogen bond, and replace some of the electrostatic functions of, let's say, a magnesium cation. I consider this an audacious experiment because I really did not expect it to work, but we had a new rotating graduate student, and these are the type of things that, well, perhaps a naive student might tackle. The system that we wanted to apply this to is an enzyme that we had been studying for several years. This is mouse endonuclease G. Our interest in this enzyme is that it recognizes and cleaves at sites that are modified by the epigenetic marker hydroxymethylcytosine, but only in the context of four-stranded holiday junctions. Mouse-EndoG, actually the catalytic mechanism of mouse-EndoG is pretty classic for a magnesium-dependent nuclease. There is a histidine, which in its unprotonated state will deprotonate of water to make that water a better nucleophile to attack a phosphate of the DNA backbone. And the DNA backbone becomes more susceptible to this nucleophilic attack because of a magnesium that can coordinate to the phosphate oxygens. And this magnesium is further coordinated to the protein itself by a standard glutamic acid. So the experiment I proposed to this graduate student was to take mouse endonuclease G, again, a magnesium-dependent nuclease, and replace the glutamic acid at position 136 with, again, a meta-substitutive of tyrosine. The idea is that this large, bulky aromatic group would displace the magnesium, and if the halogen can form a halogen bond that could electrostatically replace the magnesium's electrostatic properties, perhaps the halogen bond can also make this phosphate backbone of the DNA susceptible to nucleophilic attack. So here's the results of those first sets of experiments. We have the DNA substrate, which is both duplex and also four-stranded junction, which is the preferred substrate. When we put in the wild type mouse EndoG with its magnesium cofactor, we see that we cleave to smaller products, not surprisingly. And it's very specific in the cleavage pattern as it's associated with the 5-HMC, 5-methyl-hydroxymethylcytosine marker. Well, when we made the iodotyrosyl and chlorotyrosyl analogs of this enzyme, what we see is that, in fact, those halogenated enzymes also cleave DNA and create products that are basically identical to that of the wild type. The control study, of course, is to make the non-halogenated tyrosyl mutant, and in this case, this control experiment demonstrated that without the halogen, the enzyme is basically dead. And that tells us that the catalytic function of these two genetically expanded enzymes are associated directly with the halogen itself. Now, because the iodine enzyme is more active than the chlorine, this implicates halogen binding as the catalytic function of these new types of enzymes. We want to ask then, is in fact this activity associated with these halogenated enzymes associated with perhaps residual magnesium in our preparation? So we did an experiment where we took the iodo enzyme and the wild type and compared their activities under different levels of the chelator EDTA and at different reaction times. What we find is that as for the wild type enzyme is basically dead, does not form any cleaved products in the presence of 2 millimolar or more EDTA. whereas the iodinated enzyme remains active even up to up to 4 millimolar EDTA presence. And this could be seen in terms of just the products made, and this could also be seen in terms of the apparent catalytic rates for these enzymes. That is, the wild type basically becomes inactive up to 2 to 4 millimolar EDTA, whereas the iodinated tyrosine remains fairly active even up to 4 millimolar EDTA in solution. Now, next, so basically, these halogenated enzymes are, quote, metal-free. That is, they are very much less susceptible to EDTA's inactivation. In our next study, we looked at what the pH dependence of these halogenated enzymes look like. And what we find is that the iodo enzyme follows a classic, what I would consider a general acid catalysis profile with a pKa somewhere around 7. If we compare that now to the pH profile of the wild type enzyme has 2 pKa values. The first pKa value, again, around 7, but in this case, it serves more as a general base and would be consistent with the histidine deprotonation that can then make a water be a better nucleophile for breaking the DNA backbone. The second pKa we interpret as being equivalent to the isoelectric point of the enzyme itself, in that under acidic conditions, the enzyme would be positively charged, and as we deprotonate, the enzyme becomes negatively charged and therefore would not bind the substrate as well. Question is, why do we have this general acid catalysis with the iodinated enzyme? We attribute this to something that we had discovered a while ago called the hydrogen bond enhanced halogen bond, or the HBEXB. And this can be demonstrated by looking at the electrostatic potential maps that are calculated for, let's say, these types of models that mimic the meta-ionated or meta-halogenated tyrosines. In this case, where the hydroxyl group of the tyrosine is either pointing away from the halogen or pointing towards the halogen to form a hydrogen bond. And these electrostatic potentials show that as that hydroxyl group gets rotated towards the halogen to form a halogen bond, the electrostatic positive potential of the sigma hole becomes greatly enhanced. And we believe that this enhancement is what's forming the interactions that can help cleave the DNA. The question then is, what is the hydrogen bond donor to form these enhanced halogen bonds? Well, we looked around at the structure of the enzyme and did not find anything that would be associated with a pKa of 7, which would normally be a histidine. We did, however, find that histidine 104 is interacting with a asparagine, and this asparagine is in itself close to the methyl group of the halogen of the tyrosyl residue. And so our motto is that under high pH conditions where the histidine is deprotonated, the asparagine serves as a hydrogen bond donor to that histidine. However, if we were to add protons and protonate the histidine, the asparagine would become a hydrogen bond acceptor, which then allows the amino group to become the hydrogen bond donor to the halogen. To test this model, we started off by looking at the both the asparagine and the histidine mutants to alanines in the iodinated tyrosyl enzyme. What we find is that mutating either of these two alanines effectively kills that enzyme. However, these same mutants in the wild type remain active. What that tells us then is that asparagine 103 and histidine 104 are important, but they are not essential to the activity of the wild type. but they are completely essential for the halogenated enzymes. So what is the actual chemical cause or foundation for this catalytic function? So again, we can calculate, as the previous speaker had talked about, the electrostatic potentials, and we're going to compare the electrostatic potentials, for example, of the chlorinated and iodinated enzymes relative to their activities. What we find is that the electrostatic potentials calculated right around where the halogen is very similar to what we see in terms of the differences in the activity. That is, the tyrosyl ESP is about 10 times greater than that for the chlorine, and the activity of iodine is greater than, about 10-fold greater than the chlorinated. However, if we look at the ESPs of the iodinated relative to a magnesium and its activity, we see that the ESP differences are about 1,000-fold, not surprisingly, because magnesium harbors a formal positive 2 charge, but the activities are only different by about 1 to 10-fold. So it cannot be that electrostatics is driving the catalytic activity. What we can say then is that electrostatics can tune the difference between the iodinated versus the chlorinated, but it's not the primary driver of the catalytic function. So what is the other property that could be driving the catalytic function? Well, in addition to charge, metals also form covalent coordinated bonds. And in this case, a covalent coordinate bond, let's say between a magnesium and a water, involves the contribution of both electrons from the oxygen to a bonding sigma orbital, which would be the covalent coordinate bond. Well, what is the analogous bonding in, let's say, a halogen bond? Well, the halogen bond we had described primarily as an electrostatic interaction through the sigma Ho model. But in fact, there's another component to that, and that other component is charge transfer. In fact, the halogen bond was originally called a charge transfer bond. And in a charge transfer bond, what we see is that the electrons that contribute to the bonding orbital here, that's formed from the lowest unoccupied molecular orbital of the halogen and the highest occupied molecular orbitals of the oxygen. Again, they both come from the oxygen itself. So what we would like to propose is that in addition to electrostatics, perhaps one of the primary drivers of this catalytic function associated with halogen bonding has to do with the similarity between the covalent coordinate bond of the metals and the charge transfer bonding associated with halogen. So in summary, what we've done is we've created now a new type of biological catalyst that does not exist in nature. It is a halogen bond, and we call this a catalytic X bond or halogen bonding type dependent enzyme, or a cX-Zyme. And that these types of catalysts could be interesting in synthetic biology. We now know the mechanism, parts of the mechanisms of this catalysis is reduced metal dependence based on magnesium systems. It is, in our sense, associated with these hydrogen bond, enhanced halogen bonds. And we believe that covalency is the primary driver of catalysis, whereas electrostatics provides tuning. And as a teaser, what we've done recently is in this different system, we've been able to take a system that is simply a DNA binding pocket, and by adding a single iodine onto one of the amino acids, we can actually make a non-catalytic DNA binding pocket into an active nucleus. So perhaps a more direct and broad question is, what does it tell us about the periodic table for metals in biology if we can take a magnesium, the functional magnesium on the left-hand side of a periodic table and replace it not only functionally, but also in terms of this chemical properties by halogens, which are on the complete opposite side of the periodic table. So, if we were to look at this, we could say perhaps metals in biology are not as unique as we think they are in terms of its ability to catalyze biological reactions. These studies, I should tell you, have not yet been published, but we are preparing them for publication. And the reason they're not published yet is because of a lesson I learned from my faculty mentor when I was at OSU. That is Ken Van Holt. When we were in the process of solving the structure of the Holiday junction back in 2000, or 1999, he said, basically, make sure that your science is correct. Because if your science is correct, then yeah, you might gain some notoriety. But if science is not correct, you will certainly gain notoriety, perhaps not in the best way. To end, I want to acknowledge the people who have done the work that I've talked about today. It started with a graduate student, Gustavo Mendez, and it's been contributed by Margaret Walker, another graduate student in my lab right now, and technician who has moved on. We have collaborated with Ryan Mell at OSU to help us develop the expansion systems, and Professor Tony Rappe at Colorado State University, who helps us with our quantum calculations. And I thank you for your kind attention. And of course, funding from the NSF.
Thank you so much, Shing. That was really fantastic and fascinating, and so great to hear how far the project has come since the early days. So we can have some few minutes for questions. Ryan, you want to ask a question?
I do. Shing Ho, great to hear the story. And I don't want to take up too much time here, but we can come back to my other questions. But my first question set involves two questions. One, everything that you've done thus far has been focused almost entirely based on rational design in order to understand how this works. Have you considered yet moving to an approach where you computationally design a site, or at least build a library around your potential halogen bond site in order to enhance or discover how to enhance that kind of chemistry?
Yeah, so the answer is sort of. And so one of the problems is that computationally, if you use classic molecular dynamics type of approaches, those force fields in those classical Newtonian physics systems cannot predict halogen bonding because of the asymmetric distribution of charge. Classic MD calculations assign uniform charges to atoms. So we have developed a force field for halogen bonds, and it is making its way into those MD simulations.
That makes perfect sense. I forgot about the fact that all of those new force fields have to be built for the new amino acids. Very true, very true. Right. My next question was, you obviously have pointed out the importance of the hydroxyl on the halogenated tyrosine. And one thing that I don't know if it's available yet, but have you considered maybe taking that hydroxyl and switching it to a thiol, right? So you have a halogenated sort of thiol or a amine, and identifying whether that hydrogen bonding alteration or electrostatic alteration from the hydroxyl might alter the ability to tune the halogen strength.
Yeah, so one of the things that we don't believe that the hydroxyl group is directly contributing in terms of the hydrogen bonding in this particular case. We've seen that with the T4 lysozyme. So the first thing we're going to do is get rid of the hydroxyl group, make it just a meta halogenated phenylalanine. Just as, in fact, is the hydroxyl group contributing. And yet the other types of substituents would be interesting as well, because then that would have inductive effects in terms of tuning the strength of that signal hole and the halogen bonding potential. So that's a great question.
I do believe, just so you know, I do believe we have some some new synthetases that should incorporate those meta substitute halogens without that tyrosine, if you'd like.
Yeah, and that would be great. But there are publications that says that you can use there's a synthetase that will also incorporate the halogenated phenylalanines in a sloppier system. But yeah, it would be great if you have that working for it. We'd love to collaborate again. Start that collaboration that started in 2006, right?
Great. Any other questions from the audience? Yogesh.
Hello, Professor Ho. It was very nice talk. I'm just trying to understand, like, you have mentioned charge transfer happening from the ligand to metal. So in that scenario, if you go across the halogens, so which halogen effectively have more charge transfer as if you go across the chlorine to iodine? So in that terms, like to enhance the rate of the enzyme catalysis.
So I guess I didn't quite understand.
I'm sorry. Okay. So I'm just like trying to understand, like in the charge transfer, which halogen ideally, like if you go from the chlorine to iodine, which halogen has effective charge transfer along with the electrostatic interaction with metal?
With metal, no, we're not saying that there's charge transfer between the halogen and the metal, because there is no metal in the system anymore. What we've done is we put a bulky amino acid in there that actually displaces the metal, and basically what we are proposing is that the charge transfer between the halogen and the oxygen is mimicking a covalent coordinate bond between a metal.
Oh, I got it now. Yes, yes.
That we have this mixing of those systems because we don't. So
right.
There is covalency, and that covalency may be what is driving or allowing the halogen to make the phosphate group more susceptible to nucleophilic attack.
Right. Okay, I got it. Thank you very much for explaining that. Yeah. Thanks.
Great. I think we can do just one more real quick question from Chris. Would you like to ask a question?
I'd like to thank both of the speakers today. In particular, I really appreciate learning more about the halogen bond. And from a pharmaceutical perspective, many drugs are in fact halogenated, and I hadn't heard such a insightful description of that interaction. What my general question is, what do you think the contribution of the halogen bond to drugs and other pharmaceuticals is, and perhaps is that underappreciated?
I think it's been growing in appreciation. If you look at the top 25% grossing drugs on the market, they are halogenated. And there was a study done from a Chinese group that looked at, in fact, the pipeline for drug development. And what they found was that the starting materials, the first step, many of the compounds that start off in the pipeline are halogenated, including fluorine. And as we go through the pipeline, it is these heavy, these compounds that have iodines and bromines and chlorine, they are the ones that persist through that pipeline. And so if we start to think about drug design, in fact, the pharmaceutical companies are starting to now incorporate things like the force fields that we and other folks have developed into the molecular docking programs, into molecular dynamics programs, with the recognition that you have to now consider not only the lipophilicity, the effect of these halogens on lipophilicity of their compounds, but also in terms of providing the specificity for these compounds. For example, the one inhibitor of the protein kinase that I showed at the very beginning, if you take one of those bromines off to remove one of those halogen bonds, that inhibitor actually ends up losing 1,000-fold specificity for that particular protein kinase. And that's how important I think these halogen bonds are in drug development. In my lab, we could have gone in that direction, but we decided to go in a completely different direction, which is in terms of biomolecular engineering. But there are many people who now appreciate the need to consider halogen bonding as a important aspect of drug discovery.
And Shing, don't you have a review in 2017 about halogen bonding in medchem?
Right. So I have a review in JMedChem that has been well received that talks about halogen bonding in the drug discovery field. Thank you, Andy.
Well, thank you very much, both of you, for and being organizers of this symposium. Thank you.
Okay. Well, thank you.